In this video, we show you how to perform a plasmid isolation. We start with an E. coli culture that was grown overnight. As you can see, the solution is turbid. We centrifuge it for 10 minutes at 4000 rpm, which pellets the cells. After centrifugation, the supernatant is clear. We discard the supernatant and resuspend the pellet in a resuspension buffer, mixing truly by pipetting and vortexing. Next, we transfer the suspension to a new tube. Once we're suspended, we add the lysis solution to lyse the cells. After 1 minute of incubation, we stop the lysis by adding a neutralization buffer and mix by inverting the tube several times. Then, we let it incubate for 2 minutes. During centrifugation for 5 minutes, the cell debris forms a white pellet. The plasmid DNA remains in the supernatant, which we transfer onto the plasmid isolation column and centrifuge again. We discard the flow-through and wash the column twice with a wash solution. At this point, the plasmid DNA is bound to the column. To elute the DNA, we add millicube water to the column. After incubation, we centrifuge it. Reload the eluate onto the column and centrifuge once more. Finally, the plasmid DNA is present in the flow-through, collected in the Eppendorf tube. The column now can be discarded.